From Microbe TV, this is TWiP, This Week in Parasitism, episode number 100, recorded on January 7th, 2016. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
And Daniel Griffin.
Hello, everybody.
And congratulations on 100 episodes, Dickson. Now, you and I have been together for 100. How many have you been for? 20?
20. I started at TWiP at 80.
Are you going to be here for the next 100?
I hope so. Keep my fingers crossed.
I did mean to say good morning, Griffin.
If you... you might leave, in other words. You could go somewhere else.
No, I'm going to eat right and exercise and try to be around for 100 more episodes.
So this was supposed to be a special episode. Well, it is, right?
It is a special episode.
We had plans to do something even more special, but they fell through. Maybe we can still do that at another time. But
we could.
The fact that we're still here is special.
That's very special.
And I just wanted to tell people that through episode 99, we have had 900,000 downloads of TWiP episodes. That's about 9,000 per episode average, the range being between 4,000 and 15,000. Now, that's pretty good for a podcast, you know.
I'm flattered and honored and happy. What was our most viewed podcast? And out of the 100, which one did everybody like the most?
The most downloaded, right?
I know that number 2 was the first episode.
Right.
And I can tell you, I can pull that up in a second here. Tell us something interesting. What's the weather like?
It's not bad out there, actually, but it's warming up again.
It's freezing. It's freezing.
It's above where it should be.
This week has been cold.
I know, but we're in a warming...
You got a new band for your watch.
6 degrees on my Apple Watch. That's Celsius for those of you.
Right.
Do you like your watch, by the way?
I do. I continue to enjoy having an Apple Watch.
All right, so let's take... I'm at the stat page. We have 901,817 all-time downloads. And you know what, Dickson? The more we publish, the more downloads we get.
Is that right? There's a correlation there. Huh?
Now, the most downloaded episode is number 22, Hookworm.
Wow.
15,304.
Nice.
Number 2 is episode 1, Introduction to Parasitism, where we explain why we're calling this Week in Parasitism.
Wow.
14,700. And number 3 is Strongyloides. Number 4, Cryptosporidium.
Look at that.
Number 5, Ascaris. So all the basic ones are way up there, but they are the oldest also.
Right.
And views accrue, as they say.
Yes.
Well, that's what you like to know. Geography? You want to know what country
Sure.
has downloaded the most episodes? It's very interesting, these
Why
stats.
not?
We pay for these stats.
Well, we should broadcast them then.
We can do by geography. We can do... traffic sources, technology, etc.
Right.
Like whether you're on a laptop, a cell phone. Actually, that would be very interesting.
That would be very interesting.
All right, United States, 71%.
Yeah, as you might expect.
The UK is 5%. Other. So in the rate of listeners, US, UK, Canada, Australia, Germany, Sweden, Spain, China, Netherlands, and other regions.
Right.
As well. So let's look at technology. Let's see how people are consuming TWiP and not being infected, right?
Exactly.
TWiP 100. Yeah, we were a month since the last episode, Dickson.
Afraid so.
And we apologize. We're going to try and do it every 2 weeks, right?
Yes, we should do that. We should commit.
Well, a lot of us have schedule problems.
No. TWiP comes first. TWiP and TWIM.
If something's important, you make time for it, right?
Time for it.
Right.
So
Dickson?
this is very interesting. So the top downloader is what's called Apple Core Media, which is basically iPhone, I think. And then iTunes is next. Android is number 3. Chrome browser is number 4. Safari browser, Mozilla, Firefox, Internet Explorer, etc. So iPhone seems to dominate, huh?
I'll be darned.
And that's because there is a native app for podcasting. Many apps as well for podcast capture on iTunes.
Right.
All right. We have the usual episode for you.
We do.
And we have some follow-up from John, who has some comments on TWiP 99. 1.
Right.
At 2150, Dickson described fleas as occasional predators on humans. The standard entomological term for an insect that feeds without killing is parasite. A parasite that normally kills its host is a parasitoid. The larval stage of the star of the movie Alien was a parasitoid. If it kills several prey, it is a predator. A flea predatory on humans belongs in a science fiction horror movie or downwind of Fukushima. Okay?
Well, I must tell you, in response to that.
He's right.
He is right. He is right, but fleas that transmit bubonic plague kill many people. A single flea can kill several or more.
Are you sure about that?
Yeah, because they continue to try to feed, but
is he right?
He is right because their
I'm just enjoying this.
Their proventriculus is blocked. They attempt to draw blood up. They can't. They keep feeding. In an attempt to do that, they inject the bacteria, and then they move on to another host, and they try the same thing until they die of exhaustion.
I just want to know if you're sure about this.
I'm very sure of this one. So by
this
this
is not
definition,
This Week in Fantasy.
No, this is not This Week in Fantasy. So the point is that this definition is accurate, but I think I would exclude if it kills several prey, it's a predator. I would exclude that one. Because here's an exception, right, with the flea itself, when you come to looking at the way bubonic plague is transmitted.
Is it still transmitted?
Oh, absolutely. It's a big problem throughout the world.
Where?
Still. India, a lot of it in India. I think Dr. Griffin can probably add to this.
You know, it's interesting just to bring it here at home. We see about a dozen cases of plague every year in the U.S. The fleas associated with prairie dogs.
Okay.
Nosophylla. That's the genus Nosophylla.
So southwest U.S.?
Yeah.
Exactly.
Yeah. Well, you were in Colorado for a while. You have a lot of cases there,
and
too.
Fortunately, so people break down plague into the two types. Here we're going into This Week in Micro, but there's the bubonic plague, and the buboes basically are swollen lymph nodes. And the surgical treatment in the past is they would lance these because they become quite painful. And there's also the pneumonic plague that everyone really worries about, and that's when the plague actually spreads to the lung. Correct. And coughing, you can aerosolize, you can actually spread it that way.
That's right.
There have been a few interesting episodes where cats develop pneumonic plague. One that hit the press a number of years back in California. And this little sick cat, who got it from fleas from an outdoor animal, then transmitted it to like a dozen people through coughing.
Goodness.
So just another reason to stay away from coughing cats.
Exactly.
But no, plague's still a player in the world.
Yeah.
Number 2, at 23 minutes, you discussed how Misfly finds a mate. It is possible for a parasitic female insect to mate while all but her lady parts, as you called them, are lodged inside a host. That is how insects of the order Strepsiptera reproduce. A wingless female of the genus Xenos spends her life in a wasp's abdomen.
Wow.
A winged male flies past, smells something good in there, and has his way with her. And he sent a link to a video showing this process.
Wow.
3, at 4020, Dr. Griffin equated a cubic millimeter with a milliliter. Yes. A cubic millimeter is a micrometer.
I'm glad I'm off the hook.
So it's a cubic centimeter. It's a cubic centimeter.
Thank you for that correction. You are absolutely correct, and that was very sloppy.
And 4, at 1:15:20, you revealed that you haven't heard of famous country singer Merle Haggard. If you had, you would know how to pronounce Muskogee.
Muskogee.
And I said Muskogee probably, and everyone's cringing. And he sends a link to a YouTube video. An Okie from Muskogee. That's the way I thought it was.
Yeah.
How do you say it?
No, I corrected myself. It's Muskogee. Because it has to rhyme with Okie.
All right. Well, anyway.
Did you guys listen to the YouTube? Did anyone actually click?
I've actually heard that song sung by Merle Haggard. But
should we listen to it right now?
Just a little bit of it.
I can't because my audio is doesn't work because this is being used for.
Well, I have an iPad, so that hasn't got sound associated with it, unfortunately.
I can bring it up. I don't know if this will work. We'll see if I cause trouble.
I just want to hear this. In fact, it comes up in another email later on. Muskogee.
Muskogee.
We don't take our trips on LSD.
Oh dear.
You like that song?
You didn't ask me if I liked it or not. I'm familiar with it.
All right. Muskogee.
Muskogee.
All right. I pronounce a lot of things wrong. But I
But
don't.
they love your Italian pronunciation.
Reduvido.
When you pronounce something in Italian, there is nothing more melodic.
Reduvido is not Italian, but I can.
No, it's not. But you can make it Italian.
Dickson, che tempo fa oggi?
Oh,
Do you know what that means?
that means something to do with time.
No. And it means, what's the weather like today? See, it's
You're
very
right,
interesting because tempo is weather, and fa is from the verb to do.
Uh-huh.
So it's a weird way of expressing it, which we don't use in English. And that's often the case for other languages, right? How is the weather doing today? But how is the weather today?
Yeah, well.
And if you said fa brutto,
Fa brunto.
terrible, ugly, brutto is ugly, right?
Brutto.
Or fa bello.
Fa bello, bello, of course.
Or you could say piove,
Piove.
which means it's raining.
Piove.
Do you have enough Italian?
Not enough. but
Reduvido.
we're starting.
I pronounce a lot of things wrong, but I do pronounce Despommier correctly.
You do.
And
After
Racaniello.
years of practice.
Racaniello.
And me, I can spell your name correctly now when I email you.
And you know what's... yeah, because it fills in automatically. I went back and was editing all the early podcasts of TWiP.
Oh, my
And I used to call you Dick
You did.
until you finally said, why are you calling me Dick?
Exactly. Was it a derogatory term or were you just being friendly?
I thought that's what you were called, actually.
I-
not
actually
Dickson.
not.
So why didn't you say so before 10 episodes went by?
You never asked me.
All right, we will do a case. We had a case last time, which is over a month ago. Daniel, can you remember?
Yes, I can. I can remember, but I can read these show notes that you put in for me.
Exactly.
53-year-old woman, and she's visiting her family here in the U.S., and she came in with intense belly pain. She said it was in the right upper part of the belly. She gave us this interesting story where she had become a strict vegetarian after having been diagnosed with breast cancer, and she is on this raw vegetable diet. She says she eats carrots, local plants that she gets from the markets down there in Bolivia, we learn, is where she's visiting from. She lives in an agriculture area, and she avoids sheep, and she does not like dogs. She actually has a dog fear. She's dogophobic. That's the...
Canophobic?
Canophobic. Canidity-phobic.
I'll take a little aside there. Right? So I'm in India about a month ago, part of our delay. But and while I'm there, people are really afraid of dogs, and rightfully so. There's over 100,000 cases of rabies every year.
Right.
And I end up with some contorted travel itinerary on the way back, and I end up in Bombay, and then I'm in Delhi, and I see they have these beautiful golden retrievers, and they're petting dogs. They're comfort dogs for the tired traveler. And everybody is just a wide separation between them and the poor, lonely dogs who are just sitting there wanting to be petted, but did not seem that was the popular thing to do in India, was to pet the dogs.
You know, and if you go to the Philippines, there are signs outside of restaurants that say we serve dog here. And that doesn't mean they feed the dogs. That means they eat the dogs.
Okay.
You know what I've seen more increasingly on airplanes? People are allowed to bring their comfort dogs with them.
Yeah.
You aware of this practice?
I am not, but that seems
You
nice.
can
have to
get a
be little
note.
dogs,
You
though.
can get a note from your physician saying you need to have this dog for comfort.
Yeah. They have a size limit.
I need podcasts for comfort.
Really? You
Yeah.
get a comfort out of this? This is great.
I love it.
Okay.
Absolutely love it. I love.
He suddenly becomes
Teach
animated.
Dickson, I am a teacher by what?
Passion.
Whatever.
You have
yes.
a passion for teaching.
I just love giving people information, but not just myself. Getting experts like yourselves in to
We share
tell what
your
you know.
passion. We share your passion.
Then you should be here every day.
I am here every day. We just don't podcast every day.
Try the veal.
Okay. So
Sorry,
this
Dickson.
It's
Sorry.
okay. It's episode 100. We can say whatever we like.
You go on
So
for 3
this,
hours. That's a
Yes.
special.
Oh no, people are tuning out now. No, this woman is married. She takes care of the house, lives with her husband, and she did tell us that she noticed the pain when she came to the U.S., right? So she's here in the U.S., and then she's, boy, I'm starting to have this pain. We got a little information about the breast cancer that was localized, and it had been treated just with surgery, no additional therapy, and this was several months ago. She reported no allergies, no significant family history. She doesn't bring out any particular insect bites, but we're sure there were some insect bites. She lives in, as she says, a concrete house. This is a more substantial dwelling, not something with thatched roofs. People can think about what risks would have been there. On exam, her right upper quadrant is very tender. She has some liver enlargement. Her they do a complete blood count, some lab work. Her white count is elevated, and it is mostly, I believe it was 80% eosinophils. She has some elevated liver function tests. Her alk phos was normal. Her neuroexam was unremarkable. And she did say that before she embarked on this raw vegetable diet, she had a normal diet. She ate some meat, drank milk, but that was several months before.
All right. Daniel, can you take that first guess from Robin?
Sure. There's one of those words in there that I'm going to mispronounce, perhaps. But so Robin writes, the euboxic LFTs indicate something that isn't parenchymally invasive or disruptive in spite of the hepatomegaly. So does the minimally elevated white count. The eosinophilia goes with Naacp, in this case, parasites. He gives a reference here to Echinococcus.
That's his guess, right?
Yep.
That's his guess.
LFT's liver function tests?
Yeah.
Correct?
Yes.
Well,
Which is a
reference.
misnomer, right?
No, I don't know what that means. You don't. That's no clinical significance, Daniel, does it?
What is the NAACP abbreviation?
Well,
it kind of kills me, all the abbreviations. I think we've talked about this.
It used to be the National Association, American Association for Colored People.
It is the National Association for the Advancement of Colored People. I do not think that is where he's going.
I hope
not.
not.
Are African Americans have eosinophilia? Is that what he's saying?
I'm not sure what he's...
We don't know. It's impossible to guess.
All right, so his guess is Echinococcus.
Robin, next time clarify for us.
Listen, Dickson, can you take Jeff?
I can. Jeff writes, Dear esteemed TWiP doctors, I'm going to hazard a guess that the 53-year-old woman has chronic obstructive fasciolitis. The mode of infection isn't as clear as it could come from aquatic plants or the washing of vegetables with contaminated water. The incubation period between active infection and chronic/obstructive phase lines up well as the liver tenderness and eosinophilia. Also, this is endemic in rural Bolivia. And he cites a reference for that. Love the Twix podcasts as we regularly discuss them over lunch or in the lab. From Jeff. Thank you. So fasciola, I presume he means fasciola hepatica.
But the disease is fasciolosis.
Fasciolitis, basically.
Is that it?
Yeah,
so
that's the way
What
it
is
should
the
be.
difference between fasciolosis and fasciolitis?
The spelling.
No, you don't think fasciolosis?
No, it's the same. It's the same thing.
What is an Osis versus an Itis, Daniel?
So, the classic time we discuss this is when people have diverticulitis versus diverticulosis.
Got it.
The distinction is the itis is a degree of inflammation.
Right.
The osis just refers to some pathology.
Got it.
Existent.
All right, so next time I say that you have pathology, Dickson, I'll refer to it as an Osis.
Would you please?
Despommier Osis.
Osis.
Right.
Yes.
That's right. Not itis, unless, of course,
Well, you could have an infection.
I have advanced to another level.
Wink writes, another great case. Well, TWiP trio. Lacking Dickson's knowledge base, I had to hit the books again, but I think the Bolivian vegetarian with RUQ right upper quadrant discomfort, hepatomegaly, and peripheral eosinophilia had fasciolitis. Oh, another one.
Interesting.
Fasciolitis, or a liver fluke, and I wonder if she had ingested some aquatic vegetation from the Altiplano. I look forward to the case discussion in episode number 100. Congrats, Wink Weinberg in Atlanta. And we learned on TWiP last week that Wink has written a book on infectious diseases.
How about that? Nice.
Yeah. So here we have fasciolitis.
Right.
So now we had Osis, Itis, and Iasis.
Iasis
Daniel,
is
what's the
the-
Iasis?
That's a question for me, actually.
It is?
Yeah, because it means that's the diagnosis of what you have.
Okay.
It just refers to the organism.
Yeah.
Daniel, you are next.
Tim writes, hello, my favorite podcasting doctors. I just finished up listening to your latest episode conveniently at the same time I finished evening chores, so I don't have to come up with some odd jobs to do to stretch work a little longer to hear the rest of it. I have no idea what the cause of the case this week is, but would venture a guess of entamoeba histolytic, based on liver pain, but no animal contact, potentially ruling out fasciola, although who knows who or what pooped on her vegetables before she ate them. As the again, we Muskogee. How are we pronouncing this?
Muskogee.
Muskogee. Yeah. As for the pronunciation of Muskogee, an old country song by Merle Haggard should clear that up. And we get the link to YouTube there.
Right.
It's currently abnormally warm and muddy for this time of year, making farming a bit annoying, as I prefer to only slog through the mud in the spring when I know it will end soon and the world will green up. Winter mud is just a depressing reminder Mother Nature rules my life more so than I hers.
Oh, you don't even get close to ruling her life.
We do not.
Tim, is a farmer in Minnesota.
Nice.
I wanted to ask you something here. So we've had 3 different guesses so far.
Actually, 2 different.
2 really.
The first 2 were fasciola hepatica. And the next one was Entamoeba histolytica. Right.
Echinococcosis.
Oh, actually, that's true. We had Echinococcus. No, that was the first one, right? Wink suggested Echinococcus.
Okay. I take it all back. You're absolutely right.
All right.
You're absolutely right.
Can you take Joel's, Dickson?
Sure. Joel writes, "Hello. I have been listening to TWiP for a while now, but this is my first time writing in. It is rather warm today in Cincinnati, Ohio, at 55 degrees Fahrenheit, 13 Celsius, and actually mostly sunny for a change. I knew practically nothing about parasites before starting to listen to the podcast, so I am learning a lot, and it's really sparked an interest. As a newbie in the field, I may be way off in my guess, but after searching around, I have come to the conclusion that it is fasciolitis caused by a liver fluke. My reasoning being that it is largely endemic in Bolivia. It can be transmitted through ingestion of several types of plants or untreated water. Symptoms can include abdominal pain and liver problems, and fecal tests are not always able to detect it. I look forward to your next podcast to see if I'm right. But if not, then at least it was a learning experience. Thanks for the very informative podcast. P.S. I'm hoping that Santa brings me a copy of Parasitic Diseases for Christmas. Since it's no longer for sale, I think Santa might have failed on that one. But we can arrange something.
I sent him one.
Thank you.
I figured I'd take care of you.
That was great. That was absolutely great.
I really like that we get people thinking and looking up stuff.
Oh, absolutely.
That's really good.
Absolutely.
All right, here is Elise. Dear TWiP trifecta, how are you? Well, we are all well, right?
Yeah, we are.
It is truly unseasonably warm here in Lower Manhattan.
See?
Currently 54F, 12C. This is weird, but there is a lot of weird going on in the world right now. This is a month ago, basically, when it was warmer.
Yeah.
And just in the past week, it chilled here in the New York metropolitan area.
It did.
Didn't it?
It did. It went down below 0, in fact.
I have a guest diagnosis for the Bolivian woman visiting her daughter and suffering from upper right quadrant belly pain. I suspect she has found herself harboring Ascaris lumbricoides worms.
Oh, my.
Often in people with mild to moderate infestations, there are no symptoms at all, so it makes sense that she would have had no real symptoms until the pain in the vicinity of her liver. I know she avoids animals, but since she is on a raw vegetable diet, how is she getting enough protein? She could easily have purchased vegetables that were tainted with Ascaris eggs from the fecal matter of the local dogs she avoids, among other possible vectors. So if she didn't wash them well enough, she may have found herself some Ascaris eggs there. According to one excerpt of an article I read from a study in Alexandria, arugula is a particularly popular plant for Ascaris. One interesting note that I found about Ascaris in Bolivia is that apparently there is some recent research that suggests that infections with a lumbricoides may aid fertility. The worms may lower women's immune responses and make their bodies more hospitable not only to the worms themselves, but also to the much larger parasite. That is the growing baby. Would you call that a parasite, Dickson?
It certainly behaves parasitically, but I would not classify human embryos as parasites. Sorry.
Well, when they're teenagers, they are, though.
But that's another story entirely.
This information hit the mainstream media rather recently, and it could be dramatic puff pieces, but I thought it was interesting since the work was done in Bolivia. So she sends a couple of links. So, yeah, this came out about a month ago.
Yeah.
Not clear at all if there aren't confounding factors here. Did you see these?
Yeah, we spent a time discussing. I was not particularly impressed.
We didn't pick it as the result of its softness of hard data.
Anyway, I'm not positive of this diagnosis because in part the patient's symptoms are not gigantic blinking arrows pointing to one parasite, but given her eating habits and the fact that she lives in semi-rural Bolivia and that Ascaris infestations often have no symptoms or minimal ones, I feel comfortable. I hope the holiday season is not getting you all down and that you have adventures and intrigue and more TWiP planned for the rest of 2015 and 2016. Thank you so much, as always. So now we have 4 guesses.
4.
Did you have a good holiday, Dickson?
I had a great holiday.
You went to Costa Rica, right?
I went to Costa Rica for 2 weeks. Yeah, I had a great time.
Where did you go, Daniel? Nowhere, right?
I was up in Boston, Cape Cod a little bit.
I was right here.
But,
you were right here.
Right here, where I'm happy to be.
There you go.
Daniel.
Kurt writes, I suspect the patient harbors an unsegmented flatworm known as the liver fluke. My basis is primarily epidemiological. She lives in a rural area with livestock nearby. She ingests raw vegetables, some of which are foraged locally. If she foraged any from a stream in particular, it seems probable that she did not wash the plant, possibly assuming that the stream had done the work already. And we already know she didn't cook those plants. This is well known as problematic to savvy foragers who go looking for the likes of watercress. The elevated liver enzymes and abdominal pain with the CT being the giveaway test all but nail the coffin shut on this case for me. I'm not sure how to treat it. I'll look that up soon. Thanks for the great work, you guys. I always feel a little like Alan Dove when a new TWiP shows up, in that it's good to be here.
It's good to be here. That's what he says.
Good to be.
Oh, that's nice. I'm glad you like your TWiP. And the last one is from Arthur.
Right. Arthur writes, hello, TWiP trifecta. This is the first time I have emailed in, as I have only recently made the fabulous discovery of your podcast. As it happens, I was listening to TWiP 99 whilst feeding and cleaning the colony of Galba truncatula we keep at work, many of which are host to the intermediate form of the parasite I believe to be responsible for this worm's upper right quadrant tenderness, Fasciola hepatica. Now, the reference to Galba truncatula, what kind of an animal do you think that is?
Is that a snail?
It is a snail, because all trematodes require snails as intermediate hosts.
I listened when you talked years ago.
Oh, and you know what? You've got a better memory than I do, because I couldn't answer that question if I were asked.
Many different kinds of viral replication schemes are there?
7.
There you go, Dickson. I'm being nice to you. In this
we don't forget the essentials. That's true. But this is an odd fact to remember. That's fantastic. The clinical presentation of mild hepatomegaly is consistent with fasciolitis, as is the highest eosinophil count. However, the thing that made this diagnosis the most likely is the unusual diet the woman has currently insisted upon. Liver fluke is transmitted by the ingestion of metasicariae, which have insisted upon water plants after motile sicaria are shed from the snail intermediate host. The most adequate way to destroy the metasicariae is to either boil them or to dry them out. However, since she was not cooking the plant material, she will have been ingesting entirely viable cysts. The timing is pretty consistent with the life cycle of F. hepatica, as it takes somewhere between in the region of 3 months post-ingestion for the liver fluke to reach patency. That means to start to produce eggs, though they are likely to cause symptoms sometime before this due to migrating newly excisted juvenile fluke. Feces samples are clear, which is consistent with what we see in veterinary fascioliasis, where a number of tests are required to determine infection, since fecal egg burdens are often low. It is likely that the endemicity of this parasite is being sustained by the local ruminant population, since she lives in an area of agriculture. Thank you for your amiable chat, and keep up the fantastic work. Sincerely, the Arthur the English parasitologist. That's great.
Are you patent, Dickson?
I'm sorry, what?
Are you patent?
Say that again?
Is it patent?
Patent.
Are you patent?
Patent means that you start to produce stages.
I'm asking you, are you patent?
I haven't had a sperm count lately, but I might be. Now, you have to be shedding something that's alive.
I'm trying to get you to say no.
Okay, no.
All right, those are our guesses.
I don't even have any patents.
You don't?
No, I don't own any patents.
Good for you. What would you have patented?
That's a test for trichinosis. I actually had that on them.
So, Daniel, what else could we do to nail this diagnosis? We have 4 different guesses here,
right?
The majority of which are fasciola, right?
They are.
And what did we do? Any other tests on the woman?
We did. And,
good.
Before I tell you what that test is, I'm gonna go through what we know that might steer us. It's actually, I like this. We have a number of different options here. We actually have a true differential. People listed a bunch of things.
Are we allowed to ask questions also? Because
you're going to definitely. I definitely want to let you. I'm going to give you a little more information, but I'm also going to highlight some of the things that we know about.
Please.
So let's see the things that people brought up. They brought up echinococcus, right? That's the high data.
it's in liver.
It's in liver. So that was a good thought. They brought up ascaris.
They did, but we'll come back to that.
And the way ascaris would cause a problem in the right upper quadrant would often be they block the biliary duct, right? A worm tries to get up and blocks it, so you could end up, so that's reasonable too. Fasciola, you're in the right area. She's got the right sort of exposure history. She's got the right upper quadrant pain. This is something that actually really just tracks through the liver, so there's different ways that we might decide whether or not it's that. There's entamoeba, which again, she's in the right area of the world. It can cause a right upper quadrant pain. It can cause these extraintestinal manifestations, the amoebic liver abscess. Now, one of the clues I think we already have is we have eosinophilia. We have 80% eosinophils. And that actually is somewhat discriminatory among the potential things here. We tend not to get eosinophilia, I'll say, with echinococcus, right?
That's true.
Now, ascaris, when you get eosinophilia, it's during a certain part of the migratory phase,
This
right?
is true.
I mean, ascaris, we always say, is that lost teenager. It starts in the intestine, and then it leaves the intestine, ends up in the lungs. It finally goes back. Where does it end up? In the intestine, where it started, right?
It got to the liver even before that, too. So it's made a lot of sojourns into unknown territory, but for good reasons that actually add up to the life cycle. But I would liken it to someone that's given a grant to travel. Maybe not an errant teenager.
What did you rule out with eosinophilia again?
So I was saying echinococcus, you're not going to see
Yeah,
eosinophils.
You're not going to see.
Do you get eosinophilia with trichinosis?
Absolutely.
Yeah.
But it doesn't go to the liver, though. That's the point.
Yeah, exactly.
The other thing is you have to account for the pain in this as well.
Yeah.
There's a pain factor here that's quite serious. So echinococcus doesn't elicit any pain whatsoever. And for that reason, it's cryptic. And remember, I described a soccer player's kind of syndrome in South America, where a lot of young soccer players get kicked, and that happens all the time.
Right.
If they get kicked in the wrong place and it ruptures the hydatid cyst, they sometimes die from anaphylaxis.
Yeah.
So
Got it.
So they didn't even know they had it. They can actually go out and play soccer without ever realizing it. This woman could never have played soccer with that pain. She wouldn't have been able to do it.
Okay.
But there's more. I'm sure there's more.
There is more. So we were talking about the only time you'd see it during ascaris is during this acute, we call it Loeffler syndrome stage, when it's actually migrating through. Once it's made it back and it's kind of a chronic, and you've got adults that might lodge in the biliary tree, it might cause a right upper quadrant pain, you're not gonna be seeing the eosinophilia.
Can I add to that too? Because if are you going to come back to ascaris?
Why you go? Jump in.
No. Well, I don't want to spoil. I don't want to spoil everything in terms of the clinical picture. But I was always taught by starting with Harold Brown and then others with similar experiences that if a single ascaris worm migrates into the biliary duct and blocks it, you're in deep trouble. I mean, that's a serious emergency medicine problem. Immediate. I've never heard of an episode where the worm actually made it up into the liver and survived, and the patient survived also. I've never heard of that.
Yeah, no, you're right. It causes an obstructive, what we say, cholangitis. Yeah. And it's a medical emergency. They're going to come in right upper quadrant pain, fever. Now, when their white count is high, they're going to have something that we refer to as eosinopenia. They're actually going to clear their eosinophils because they're going to become septic from basically bacteria that are in the blocked biliary tree.
And maybe a shift to the left.
They'll get a shift to the left. They'll have bands of high neutrophils. They'll have no eosinophils.
Right.
So
that must mean that there's a crossroads in the bone marrow between making eosinophils or making neutrophils, which is quite interesting, I think.
Yeah. And also there's the issue that steroids themselves seem to have an effect on clearing eosinophils from the bloodstream.
Right. So that I would rule out acute infection with Ascaris because of that.
Yeah. But I like it included in the differential. I think it's nice people are thinking of this.
Absolutely. What else could be in the liver? And that's the way to think about this.
Yeah. Right. The other thing that was brought up was the idea of an amoebic liver abscess.
Yeah. Right.
Probably not going to get eosinophilia again with amoeba. There again, you're going to get a high white count, and probably not going to get 6,000, 12 times the upper limit of eosinophils with amoeba. But how are we?
Double infection.
That's always true. But how are we going to make the distinction here of right upper quadrant pain? The classic test would be a imaging test to look at the liver, see what's going on. Are we going to see gas or something going in the biliary tree? Are we going to see a solid cyst? Or are we going to see basically destructive tracking through the liver? And I must say, we saw this wonderful liver parenchymal tracking, basically streaks, destructive streaks traveling through the liver.
Wow.
I have a tiny little picture that you can see there. I had to show it to Dickson. It's pretty impressive.
Very impressive.
And there was ELISA, an antibody test that was positive for a particular organism.
The other thing that could have been added to this as an adjunct diagnostic test would be an ultrasound. I've actually seen ultrasound pictures of livers with this particular entity in it, and you can actually see the tracts much clearer than you can on what is this, an MRI?
This is a CT, but actually you're right. We tend to do a lot of CTs and MRIs in the U.S. where ultrasound is actually an excellent imaging modality without the radiation risk and without the tremendous expense that is employed through much of the world.
And without a gigantic magnet necessary to actually make it happen.
Exactly.
About 2 years ago, we had our son had a mass on his adrenal, and they brought him in to do ultrasound.
Yeah.
And, he's there with a number of people, technicians,
Right.
And then we finish, and we go to the doc, and he's looking at the picture. He said, I can't see anything, and these are crap.
Oh, dear.
Because the guy would say, okay, here's the bladder, here's the kidney, blah, blah, blah. And the doctor's like, I don't see any of this.
Where do you go to medical school?
He said, we're not going to bother doing it again.
Oh, dear.
No, he was an experienced surgeon.
You can tell the sex of your child.
But he said the ultrasound was no good. But we're sitting there, and he's saying, look, here's this. Oh, okay. Because I don't know what's what in an ultrasound. So I don't know.
Yeah, the ultrasounds have really come a long way. I still remember when I was in medical school, and based on ultrasound, you could tell the sex of your child with about 50-50.
Exactly.
Now you really see everything, and you're like, there it is.
Oh, you can see cleft palate. You can see the development. I mean, it's remarkable.
Well, the surgeon wanted to just know what he was going to see when he went in the next day, right?
Right.
Yep.
And
so when you saw that, what did you jump out of your chair?
And I should say, and the ELISA was positive for fasciola.
Right. Did you do a stool exam also?
So we actually had talked about that last time, and I thought that was, so the stool O&P was negative. Right. And how do we explain that? And that also goes back to what we talked about with the Ascaris. When you're thinking about your parasite, you need to understand the life cycle. Right. And at this stage, the fasciola organism, Fasciola hepatica in this instance, is still tracking through the liver, has yet to find the biliary tract where it will start to get eggs and release them into the stool. So at this point, you will not find eggs on stool examination.
Right.
If you wait long enough.
Do you think this could all have been caused by a single worm?
I don't know. I'm not sure how many she was
that's another reason for not finding eggs then. I mean, if it was only a single worm, these worms, although they're hermaphroditic, they need to mate with each other. So they actually reverse themselves, and a pair of worms actually is required for egg production. So maybe that's a single worm doing all of that.
Yeah, it could have been.
How did you treat
and they're pretty big.
They're classic biology specimens for high school undergraduate or high school courses. It looks like a leaf.
Yeah.
A small leaf. And they're beautiful organisms, basically. They're actually gorgeous. But, of course, they're pathological as well.
So the treatment is great. The treatment is, so this is the one fluke, and this is always the board question, that ends up getting treated with the medicine that you don't use to treat the other flukes. So all the other flukes get praziquantel. This fluke gets triclobendazole.
Triclobendazole.
So it needs its own special drug.
Exactly.
How long?
And actually, so she was treated, I forget actually how long she was treated for, but then we did do serology actually 2 years later. She cleared. So
And
this is
how was her liver at that point? It didn't look
the liver actually returns to normal. It looks good. I mean, that's a great thing about the liver is actually the regenerative ability. And it's always amazing. It regenerates, but it knows exactly where to regenerate. So it doesn't become odd or abnormal or...
Stem cells. It's all about stem cells.
Yeah, but somehow there's a contact programming where they basically recreate the liver, and it looks great.
Remarkable.
How long after taking the drug did her pain go away?
Pain went away pretty quickly, actually.
A week?
Maybe 2, yeah.
Okay.
So this is, you'd be tempted, and rightly so, I guess, upon hearing about this for the first time, to think that, well, that's Bolivia. That's a country that's pretty poorly developed in terms of its political structure, and there's very little control in terms of public health measures in many areas, and that's kind of a rare finding that, if you just don't do what this woman did in Bolivia, you'd be fine. Take a guess where else this is found.
U.S.?
Yes.
South America?
Oh, well,
Bolivia.
Bolivia's in South
Bolivia.
America.
Elsewhere in South America.
Take a guess, like Ireland, for instance.
Really?
You really?
But it is.
Yeah.
It is in the U.S. now and then.
Yeah, it is, but it's not as common in the U.S. as it used to be. But it's present throughout the British Isles. It's present in Australia. It's present in New Zealand. So these are countries with very good healthcare systems, and they still have a difficult time controlling it.
Yeah,
So
Hawaii is the big
And Hawaii.
U.S.
The moral is cook your vegetables. Right?
Cook those vegetables. So you go into a restaurant and they serve you as a side dish, right? A salad. That's
Yeah.
not cooked,
No.
is it?
Yeah,
So-
but lettuce usually doesn't come from streams, does it? Or is it contaminated with
not necessarily
stream water?
streams. These are little ponds, for instance, that
Yeah,
have
okay.
growth around them. Now, why is kale such a popular source for this, do you think? I don't either. I mean, I'm asking a question now. Why would kale be one of the prime sources for this, for the Metasicaria? How do they grow kale? That's the point.
Well, clearly, Dickson, it should be grown in a vertical farm, and then
Well,
you get around this
I've
problem,
actually
right?
experienced that myself firsthand, and kale is fantastic when it's fresh picked. So, but I don't really know what the agricultural setting is for kale production in Bolivia, let's say, all right, where it may come from as well. And it probably obviously involves a cohabitation of that crop with sheep and lots of other animals, and maybe they even feed it to the sheep, who knows, as a nutrient. But it's a great case because I did not guess fasciola to begin
What did you
with.
guess?
I thought it was Entamoeba histolytica with a superinfection of strongyloides.
Wow. That would be
Two things. You can have two things here,
yeah,
folks.
Sure, why not?
And, there is upper right quadrant pain with histolytica, but it's mostly left upper right quadrant rather than right upper right quadrant. But nonetheless, a constant pain is unusual for a parasitic infection. A constant, low-grade constant pain it's tougher to account for it.
So maybe the case I present this week should have two things going on. Just to, I already picked it out. So.
That is reality, folks. I mean, I've been a technician for a while, and I can tell you, we had one patient that had 5 different things. I mean, 2 things on blood smear and 3 things on their stool exam. So, yeah, I mean, when you're living in an endemic center for one parasite, it doesn't mean that the others can't be there also. So that's a good thing to think about.
All right. Shall we
That was
do?
great,
shall we?
by the way.
Shall we do a paper now?
Sure.
We have a
Wait a minute.
paper.
What are you going to do about prevention? This is a big deal here.
Prevention.
How would you prevent fasciola infection?
Cook your vegetables.
That's one way. That's on an individual basis. What about an epidemiological prevention strategy?
Kill the snails.
That's another one that they've tried.
Doesn't work, right?
Well, it does, but unfortunately, it kills everything else in the aquatic environment.
Indoor farming.
Well, no, I wasn't going to say that. But how would you control it in the reservoir host? The reservoir host is the sheep. That's liver fluke. It's called sheep liver fluke. So maybe there's a need for starting off with fasciola-free sheep and introducing them that way. And then that way you won't have the problem.
Kill all the sheep and replace them.
Yeah, they did that, by the way, in Iceland to prevent the spread of echinococcus when they had a big problem with that one. So it is possible to do that.
But then you have to make sure they don't get reinfected.
Exactly right. So they have strict immigration laws about bringing agricultural products into the countries and stuff like that. Yeah. So, great stuff.
All right.
All right. This paper was selected by Daniel. It's published in the Proceedings of the National Academy of Sciences, of which none of us is a member.
That's true.
And,
or ever will be, by the way.
Probably not. Well, you never know. Daniel's still young.
This is true. This is true.
But it's fine. We are not bitter at all because
not in the least.
There is no National Academy member that has the number of listeners that we do.
That might be. I don't know.
I would definitely.
You know of any National Academy members that have their own podcast?
Although, is Neil deGrasse Tyson in the National Academy?
That's a great question.
Because he's got a lot of radio listeners.
That's a great question. He also has a TV show now. So.
Yeah. This paper is called Highly Efficient Cas9-Mediated Gene Drive for Population Modification of the Malaria Vector Mosquitoes, Anopheles stevensii. Now, Dickson.
Stevens eye.
Stevens eye?
Yes, Stevens eye. Stevens eye.
Stevens eye?
That's right. It's a normal carrier of malaria in India that's been colonized.
Now this is a very interesting paper, which is really has a lot of potential, but doesn't cure anything in the end. But the point is, this is one way to approach malaria, and there are many others, to alter the vector in some way.
That's right.
And I don't know if you remember, Dickson, but on TWiP, we did talk about one approach, which is to introduce Wolbachia, which are bacteria, endosymbiotic bacteria, into mosquitoes.
Yes, that's right.
It now makes them resistant to dengue, and then you release them. And that was done in Australia.
Exactly.
And it's quite effective at reducing dengue virus replication. What's the effect on malaria, do you know, of putting Wolbachia in?
That's a good question. I don't know.
And it's known. I should know that. But one of the main issues is getting these mosquitoes, if you're going to release them, and of course that in itself is a huge issue. And I will just say right here up front, this will never, ever happen in the world. There's no way.
Don't tell that to Tony James, who we've had on TWiP, by the way.
Yes, we've had Tony on TWiP. And I had a big discussion with my younger son the other day about genetically modified organisms.
Yeah.
And this is why, because people don't want to consume genetically modified organisms, even though they do all the time, like rice and corn.
That's right. That's right.
They don't want to do it. They want a choice. So I think people want a choice in their mosquitoes as well.
There's something else here, too. I mean, you're releasing a vector that could transmit other infections.
Better?
No, but you're just increasing the pool of mosquitoes for things other than malaria.
That's a good point. I never thought of.
So what other things do Anopheles stevensii transmit?
What is it?
Well, you could throw filariasis in there.
You would only release this where the mosquito is endemic, right?
Well, it's endemic to India. It is.
So you'd release it in India.
12% of the vector species in India are Anopheles stevensii. 12%.
Well, aside from these issues, the real issue is you put a gene in a mosquito.
Right.
And then it goes out there and it mates.
Right.
And according to Mendel, what happens? If you're lucky, if you made a homozygous mosquito, two copies of that gene,
50,
then the offspring are going to have
50%.
Then less and less. It's going to be diluted.
Correct.
So you have to have a way of getting that gene to propagate, and that's called a driver.
That's right.
And I think this is the best thing to come out of this paper, the word gene driver.
Oh, that's an old concept.
Hey, Gene, how are you? I'm
that's
fine,
right.
Driving around.
That's right. Well, what happens when they introduce driverless vectors? No, that's not true.
So you need a gene driver, or he actually points out there's one approach where you can inundate the region with mosquitoes. You could dump billions and billions.
Yeah,
so
that's
many.
even worse.
But he said, that's not great, but we want to do a driver. Now,
that's right.
So this is what's really cool here, a driver.
He likes this.
Because let's talk a little bit about what a driver would do. Okay. So basically, if you have a gene in an organism that has sex, at every generation, you can have at best 50% of the offspring with that gene.
Right.
And that's no good for this. But a driver is going to be in everybody, right? Mm-hmm. You guys agree? And I found this wonderful document, which was produced at Harvard. And they say in Nature, an example of a driver is what's called the P element. It is a DNA in fruit flies that was absent completely before 1950, but now has spread throughout all the pop. We don't know where it came from, but it drove...
Transposable element, right?
It's a transposable element.
It's
everywhere. Every sexually reproducing species has an active gene drive or the broken remnants of one in its genome.
How about that?
Now, the really cool part here is that with CRISPR-Cas, we can make a driver out of any gene, basically.
Correct.
Because if you put a gene in along with CRISPR-Cas, as soon as that's in the organism, it's going to muck with the other copy and put the gene in the other copy.
Right.
So every organism becomes homozygous for your gene.
Correct.
It can be flies, mosquitoes, whatever you want to do. So this is really cool.
So Vince, I challenge you.
You can take any species, any gene you want, and drive it in the organism.
And that's what he did.
An interesting question, Dickson, is could you do this in people? Could you drive a gene in people?
Well, they've already done it in pigs. They completely removed all of the retroviral-like sequences in the pig's genome using CRISPR.
Yeah, but have you repopulated all the pigs in the world with that? No.
Well,
that's what a gene driver does.
They have not done that.
So could we do something to a human and have all humans? No. And the reason is because it takes too long in people.
Well, the other thing is you shouldn't do it.
I don't know about that, Dickson.
You know, David Baltimore was recently quoted as saying...
Dickson, let's not make these judgmental statements, what we should or should not do. I'm not sure that we're in a position... You know, I view this as part of evolution, us being able to modify ourselves. Okay?
We've already done that by selection.
But anyway, if you could put a gene driver into a human, could it get through the whole human population?
Well, your argument is valid.
It would take too long. It would take too long.
This is probably...
And there's a wonderful example in this document. It says, for example, if we introduced a trait into elephants using gene drive today, there would be only 4 times as many elephants with that trait in 100 years.
Okay,
if we didn't use a drive. Wait, that assumes no one decided to remove the trait, which is quite possible. So it takes too long.
Right.
But mosquitoes reproduce in what, 14 days?
Less. Yeah, a week and a half.
What do you want to say, Dickson?
I wanted to ask, is there an example of a disease which is transmitted by sexual contact, which has spread through the human population rapidly? Because that's your answer to your question as to whether you can introduce a gene driver.
No, but I think it's more than that. It isn't just the spread of the disease. There's got to be reproduction involved.
This is true.
Yes.
It's got to affect the germline. So there are diseases which we can all think of that have been sexually transmitted and have spread all around.
Right.
But it's a truism that you always have at least as much sex as children you have. So the number of sexual episodes is always equal to or greater than the number of progeny. Almost always true.
Okay.
I have a question for you, Dickson.
Please.
Could you make a gene driver of crops?
Oh, sure. Absolutely.
No. The answer is, in theory, you could, but it's unlikely because
Yes.
crops are usually made from seeds that are monocultures that are made from start.
Oh, you mean could you get them to breed it into the population?
Yeah, you don't do that with crops.
No, you
That's
don't.
the problem. Similarly, cattle and most other domesticated animals are products of tightly controlled breeding programs that rely on artificial insemination. So you can't drive unless you change those practices.
Unless you change your practice. So
Yeah, you have to
what
change
we were hoping, both Dr. Griffin and myself, was, since you are the genetics expert in this crowd, although Dr. Griffin knows a lot about that as well, I would love you to, for those listeners that are unfamiliar with the CRISPR-Cas system, to simplify, without diluting the intellectual content of your explanation, your view of the way the CRISPR-Cas9 system works. Because in 2015, the last issue of Science Magazine features the molecule of the year. Last year, CRISPR-Cas9 was runner-up. This year, it won.
It won. So what does it get?
I don't know. I have no idea whatsoever. But they had a little picture on the cover of Science that sort of indicated that it's like a game of Tinker Toys that you could, or Lego. There's a combination of Lego and Tinker Toys. You can take little pieces out and play with them and then put them anywhere you want and make something new out of it. I think that's the gist of CRISPR-Cas9.
No.
But why don't you give us your simple explanation?
Let's make it very simple.
Yeah, please do.
Originally, CRISPR-Cas9 is part of a bacterial and archaeal defense system against viruses, plasmids, invading DNAs. All right, it's been
restriction enzymes are.
Yeah, and it's
Okay.
a different way of doing a similar thing, right? Restriction enzymes take a DNA that comes into a bacterium and cuts it.
Right.
Now, I just have to take a little aside here.
Please.
Restriction enzymes were studied in the 50s and 60s. Nobody knew.
Yeah, exactly.
Man,
They did.
but people in science, we were always allowed to pursue our curiosities. And out of restriction enzymes came the biotechnology, multi-billion-dollar biotechnology industry, which is now beginning to impact people's health.
Exactly.
Similarly, CRISPR-Cas was an obscure repeated sequence in bacterial genomes, archaeal genomes. Even Jennifer Doudna said when someone told her to work on it, she said, "Well, I don't want to work on this obscure thing. Nobody knows what it does."
Right.
And the point here is that you have to let scientists work on what they think is interesting, because that's
true.
where good stuff comes from. And the reason I'm bringing it up is because some of the critics of gain-of-function research have recently said this work has no merit. And I don't see how you could say that any work has merit when you can't predict what's going to come of it.
Right.
And this is a great example of that. So as part of this wonderful defense system, Cas9 is a nuclease.
It's a protein.
It's an endonuclease.
It's an endonuclease. Okay. It's an enzyme that chops up DNA. And it is the actual enzyme that will, when given a sequence, in the form of guide RNAs, they'll call, it will take and go to the gene that those guide RNAs are from and cut that gene. It will cut between the two guide RNAs. So you can put the two guide RNAs at either ends of a gene, and the guide RNAs can be produced as a single RNA. In fact, it's amazing. And then the Cas9 will cut the gene out, and then your genome will repair the gap, and now you won't have the gene anymore.
So I have a question about that.
So that's basically how it works, and that's how you can repair genes. You can cut out pieces, and you can add stuff in.
But how does a bacteria take advantage of that? It has to alter that RNA every single time.
So the bacteria has a memory of all the assaults it's had. It stores them as sequences in its genome.
Okay.
And then these are made into RNAs. And then when that sequence comes back, the nuclease takes the guide RNA and chops it up.
Sure.
And that's how they were first detected, because there were these arrays of repeated sequences. And people said, what are these doing here? And eventually they figured out that if you take them out, you take away the ability to defend. Yeah. And then someone, actually a guy in Lithuania, discovered that it was a nuclease that you needed for this protection.
I'll be darned.
And Jennifer Doudna, of course, showed that you can put these two together, the guide RNAs and the Cas, and it would do the same thing.
So can you sequence the guide RNAs from a given bacteria and determine who their enemies are?
Yeah, for sure. You get a history. It's like reading a book.
Look at that.
Absolutely. You get their history. So that's what they use Cas9 and guide RNAs to make a driver in mosquitoes in this paper, basically.
Right.
They are going to say, could we do something that would then propagate through all the offspring? So if we release these mosquitoes, if we release them, would the gene drive through the population? Because that's what you need. You don't want it to be diluted by Mendel.
Sure. No. Or Mendel, depending on how you want to pronounce that.
I'm pronouncing everything wrong today. Stephen Z. and Mendel. But that's the whole point of this paper, and it's a proof of concept.
Yeah.
It's brilliant.
Correct.
It's brilliant, and it's wonderful genetics.
Correct.
But really the bottom line is, the answer is yes. You can make a driver in mosquitoes.
So what else did they do?
Persist.
That's right. They did one other thing too, didn't they?
What did they do?
Well, they introduced a gene that carries the sequence.
So that would be the idea. You don't want to. You want to put something
based antibodies. I read this paper.
Camel-based antibodies.
Right.
Against what?
Against Plasmodium falciparum.
Right. Or is it falciparum?
It's a falciparum. And in Britain,
I like falciparum.
Fine. And they were against two different antigens from the parasite. One was from the sporozoite surface coat for the sporozoite, and the other was from some erythrocyte stage.
So the idea would be that these if these were produced in a mosquito, what would they do to an incoming malaria?
Kill it.
That would be the idea.
That is what happened.
Well, they don't even show that in this paper.
They have another paper that says that, though.
Do they?
They do. That's how I read. That's how I discovered this.
So they just show this is the proof of concept of the driver.
Right.
They want to say, can we put these two genes in a vector, and will this be driven through the offspring of the mosquitoes?
That's right. The reason why they picked the second gene for the antibody was because in a prior study they had already proven that if
in mosquitoes it would do it. Yes,
it would kill it.
But they didn't actually use this driver concept. And the next step is to show that these mosquitoes that inherit the Cas9 and the guide RNAs actually are resistant to infection.
That's right.
And then if that works, the next step would be to see if they could do a release experiment.
Tony concludes in one of his editorials that he doesn't think in his lifetime he'll ever be permitted to do that.
I agree with him. I absolutely agree with him. But the science is very cool. So I wanted to go over a couple of the points here, which are very interesting. So the idea is you make a plasmid.
Right.
They make a very big plasmid with a number of things on it.
It's big. Exactly. 17 kb is
17 kb plasmid. So on it are these genes encoding these two anti-malarial components that you just mentioned. They have
single-chain antibodies.
Right. They have a red fluorescent protein gene to serve as
a red fluorescent, not a green. That's right.
And then they have a Cas9 gene that encodes the nuclease.
Right.
Because once it's in the mosquito genome, then that's going to chop out and transpose to the other. That's how the mosquitoes will get to have both copies of the gene. That's how it will drive.
But then you have to screen.
And then, they have a sequence that targets it to a gene in mosquitoes called, it encodes the kynurenine monooxygenase gene.
Yeah, I had to write that down too.
Which deals with eye color.
Exactly. So that you can do a screen just by looking.
Yeah, so the eye disruption of this gene, both copies makes white eyes.
Exactly.
All right, and so that's what their screen is to know if they've gotten in here. Now, they take this plasmid and they put it into 680 embryos of Stephen's eye. And they're injected with 100 nanograms per microliter. Dickson, tell us how hard it is to inject a mosquito. An embryo.
Not hard. It is not hard.
An embryo is, what does this look like? Is this an egg, basically? So that's not too hard.
It's not hard to do. And in fact, one of the great researchers in malaria, Dr. Lewis Miller, who happened to be one of my classmates here at Columbia way back in the day,
I remember, yes.
Spent most of his adult life after going to NIH learning the technology to inject into mosquitoes so he can start transfections. He never succeeded, by the way, but Tony James did succeed. And do you know why the difference was between those two people?
What?
Tony James had experience with Drosophila.
He did.
So he used promoters that were Drosophila-based, and as a result, got the genes.
In fact, the first gene drivers were made in Drosophila.
To go into the mosquitoes,
so
because they share common promoters.
He applied that to mosquitoes.
Whereas Lou Miller never worked with Drosophila, and therefore never thought of doing that particular kind of experiment.
Now, there's a cool technical detail here that I wanted to mention, but it's really technical, okay? But it's cool.
Please.
So there's a Cas9 gene on this plasmid, and that is going to work later when the mosquito is reproducing, and it's going to be integrated in one copy of the gene, and then it's going to cut the other copy, and it's going to move over and duplicate. This is called gene conversion.
Okay.
They don't want that copy to be active during the injection process. They actually supply Cas9 protein, which is injected into the embryos. So they have an RNA that will silence that DNA because they don't want it to be active. They don't want it to cut itself out there. Not yet, later. Anyway, out of these 680 wild-type embryos, they got 122 and 129 males and females that survived to the adult stage.
Right.
And then out of those, 2 males were positive for DS red fluorescence.
Didn't they screen like 25,000 insects before they...
Following 25,000 larvae,
That's right. That's incredible.
After crossing. So they took those 122 and 129, and they crossed them, and then they got 2 males that were DS red positive. Right. And they showed that these had integrated the DS red into the right place. And then they basically did crosses to say, is this, in the eye color and the DS red, is it segregating Mendelian fashion or not? And in fact, what they were surprised is that most of the offspring had the plasmid.
Exactly.
Way more than you'd expect for Mendelian inheritance, right?
Exceeded all
non-Mendelian segregation patterns. So Mendelian would be 50% of the offspring would have your gene. And here the numbers are amazing. 99% of the offspring have the plasmid in them. So basically it's a proof of concept that shows that these individuals have the gene drive construct and they can pass it on. And they do several generations of breeding and it keeps going.
Can I?
Do you agree with that?
I love that.
Isn't that great?
I love it. I absolutely love it. So to go right back to the beginning,
germline. It goes in the germline.
It goes in the germline, exactly. So to go right back to the history of this, if you went back to the invention of modern genetics,
who would you say did that, Dickson?
Oh, we know who did that.
Gregor Mendel?
No,
he's not modern, right?
Gregor Mendel was a priest.
So you have a
did
problem
experiments.
With that,
No, you know who didn't have a problem with it was Theodosius Dobzhansky.
Thomas Hunt Morgan.
Well, he informed Thomas Hunt Morgan and Sturtevant and Stiles as to the significance of this.
This was the Drosophila school.
And where was that done?
Here at Columbia.
Damn right.
I know, and there's a fly room here.
You're damn right.
Why didn't we do this podcast from the fly room?
That could have been right. So take a guess how long, once they decided on their proper genetic organism, which was the fruit fly, take a guess how long it took them to find one mutant fly.
How many years?
Yeah.
10 years.
3 years of constant looking for a mutant without finding one. Because they didn't know whether or not there were any, okay? Whereas Tony James has the advantage of creating a mutant And he has a technique which makes it easy to find.
Well, his first one was not so easy.
No, it wasn't.
25,000.
No, well, but the point is, you're looking for white eyes, so you just keep zooming through them until you find white
We
eyes.
did on a recent TWiVo. We spoke with a
Yes.
gentleman who did a Drosophila screen.
Okay.
And he had an idea, as a postdoc, he had an idea to identify a gene involved in a certain trait.
Right.
And so his boss said, "This is a great idea. Do it." And he said, so they started doing the matings. He said 9 months into this, they didn't have it yet. And he's like, should I keep going? And they eventually got one mutant and then another and another and another. But he said, you have to make a decision. Are you going to keep going or are you going to quit?
It's commitment.
Because this guy's postdoc. And if
It's
he goes
commitment.
2 years with nothing, he's kind of finished.
Listen, you usually think of genetics as a rapid type of behavior, but
not at
it's
all.
not sometimes.
I mean, this paper is a lot of work. This is
and a collaborative effort with lots of geneticists and molecular biologists.
And there are many details that we can't go into.
Right.
For example, a few of the flies don't get this driver by the driver method. It goes in a different way, and it's inherited in a Mendelian fashion. But it's very small. Although in nature, those could overcome. Those could overbreed. Who knows?
It would be so below the transmission level of the density of mosquitoes needed to transmit that you wouldn't worry about that part.
Now,
so you could try this on an island where there is malaria and where you
I have a perfect island for that.
You do now.
New Jersey. Oh, it's not an island, is it?
Not yet.
I was going to suggest Long Island.
Long Island. Well, it has to have malaria, endemic malaria as well. So some small island off the coast of India might be a good place to start because Anopheles stevensii exists there already.
Dickson, they have these two genes for the single chain antibodies.
Yeah.
They have placed them downstream of a promoter that is blood meal inducible.
That's another good thing.
Why did they do that, Dickson?
Well, because they don't want. Well, maybe because they don't want to burden the male mosquitoes with the task of producing these, because they don't feed on blood. So the males would. It would be a waste of.
In fact, they say they look for RNA of these two single chain antibodies, and they show that they're turned on after a blood meal. So they're actually not just driven through the population, but the RNA is made. And the male, and they say as expected, samples from males show no expression from either transgene because they don't take blood, right?
They do not.
So this
In
thing
fact,
is
the females don't feed on blood either. Remember that. They feed on plant juices just like the males.
They take blood meals.
No, they're called blood meals, but they don't use them as food.
What would you call something that you put in your mouth and gets into your gut? It's not a meal?
Well, no, I'm telling you that it doesn't end up there.
Yeah,
there's no nutritional value.
No nutritional value. So a meal, in your view,
it has to have nutritional value.
To have nutritional value.
So. Don't you think that's
What do you
true?
think, Daniel? Do you think a meal has to have nutritional value?
I understand the distinction here that the blood meal that the female mosquito takes is used for the egg development, but we still call it a blood meal.
We do because in the past we did know better, but I think
so. Tony,
but I would understand. This is like when when your mom sees you eating a bag of potato chips and you claim it's lunch, and she says that's not lunch.
Right, it's junk meal. It's junk food.
But Tony calls
maybe
it
blood
a
is junk food.
Tony calls it a blood meal. But I'm wondering if it's needed for the production of eggs. Isn't that nutritional value?
Well, it is for the survival of the species, that's for sure.
I think we call it a blood meal.
I'm not saying we don't. I'm just
Daniel, was there anything that you liked about this paper? Not that you have said you don't like anything. I'm just
I was very quiet. No, I have to say I was really I liked the science. I liked how they did this. I liked the fact that they came up with the idea that we want to be able to verify that it's gotten into both chromosomes, and so they had the recessive as well as the dominant traits. The dominant trait is the red, the DS red.
Right.
So the DS red's there. Okay, you know it's there, but you also want to know, is it in both?
Exactly.
And that was why they disrupted this eye color gene
because it's a recessive.
Yeah, so you could look at the mosquito, the larvae, even the adult, and say, is it making the red?
Yeah.
Is it also white-eyed? Now you know it's in both. As I think we've mentioned over time, the concept of gene drive is not brand new. It's been out, for
a long time,
for a while.
That's right.
In other endonuclease systems is when we sort of discovered this. But now Cas9, we've rediscovered it, in a lot of ways, we've rediscovered it because now it's a much easier to use tool. I mean, the endonucleases were proteins that were hard to manipulate. Here, you basically can just design up your plasmid, put whatever you want in there. And in this case, as a drug, you could say, well, okay, I'm going to put this in here. This is going to be expressed after a blood meal. This is going to be only expressed in gametocytes. I mean, we can really fine tune this CRISPR-Cas9 tool.
That's true.
I thought it was pretty impressive.
So basically, you take a mosquito that has both this plasmid, which is in this case is going to produce the single chain antibodies, or it could be to do anything in any animal that has sex. It will release them. They have the plasmid in both chromosomes. That will then mate with mosquitoes out there. Then those will each get one copy, but in those mosquitoes, that copy will be duplicated by the Cas.
There you go.
Into the other. And when they mate, 50% will get it. Everybody gets it.
That's right.
And that's the title. Everybody gets it.
Everybody gets it. That's right.
But you think Tony? You said Tony has gone on record as saying he's never going to see this.
Yeah, because he realizes there's a lot of social aspects to this, and a lot of political aspects to this too. I mean, when people realize that you had cages full of a known vector for a parasite, even though you've convinced them otherwise that they can't transmit it, it's hard to convince them that they're not going to get something else as the result of this, and you might raise the level of mosquitoes to the threshold of transmitting filariasis, for instance, which you wouldn't want to do.
Yeah,
and all these mosquito control programs are all aimed at eliminating insects rather than adding insects, right? So it's got some onerousness with regards to social marketing. And if you don't social market this correctly, people will accuse you of trying to spread diseases, which you're not. You're trying to prevent them. But I think he's encountered this already once with his dengue experiments, and now
So he was involved with the dengue Wolbachia, right?
Yeah, he was. That's right. That's right.
So I'm going to say something controversial, right?
Please do.
We usually
Doctors
don't.
No, I look at this, and every year we have between 500,000 and 1 million people dying of malaria, right? I mean, we have a crisis.
That's right.
Hundreds of thousands of people dying.
Right.
And so here we have potentially an approach. I mean, I think we're in sort of a bad situation right now. And I think rather than saying it's never going to get out there, we've got to sort of ask questions. Is it really realistic to feel that this technology is going to create some other problem that would be worse? Are these mosquitoes going to somehow be better at spreading other viruses?
Sure.
Are now suddenly there going to be more mosquitoes? Because in a sense, what we're looking at is just replacing the current mosquitoes with mosquitoes that can't spread malaria.
No.
It's not going to be more.
There's no reason to think there'd be more.
Right.
That's right.
But the question is, could it be better for something else? And you should put a kill switch in them so that you could get rid of them all if it came out to be a problem.
Speaking of kill switches, wouldn't it be great that if you had a gene that was lethal for the mosquito that was inducible by the blood meal act?
But I guess we don't want that, right?
Well, then you're never
going to get it to spread through the...
Within a week
it's not going to drive through the population then.
Well,
you need something that
afterwards.
You could do something innocuous to get rid of them all. So if you sprayed them with sugar water, it would kill all of them.
Yeah, well,
if it turned out that they were a problem, that's what a kill switch is, right?
I mean, somebody had an idea that insecticide impregnated bed netting was an answer to the control of malaria, and that was really grabbed onto by WHO.
Didn't it help?
It did help. It lowered the rate of malaria in some areas by 50%. But guess what else it did?
Resistance?
Yeah. It selected for insecticide resistance.
Of course.
And now the bed nets don't work anymore.
Really?
Yeah, they don't.
I think they're less effective. I wouldn't say they don't work.
Well, in some areas they don't work. In other areas they work less well, and that's going to happen. Your evolution will catch up to you.
Yeah.
All right?
I guess in a sense
maybe that's the bottom line on this one.
I guess in a sense here, we're actually jumping in and messing with evolution.
We certainly are.
And if we can put in a gene that makes them resistant to malaria, we can put in a gene that makes them susceptible to X, insecticide Q. And so you spread this out, they can't carry malaria, and we say, but by the way, at the same time, we've created mosquitoes that are now all susceptible to this insecticide. So we put a gene in that makes them hypersensitive.
Yeah, but that gene can mutate. It does all the time, and they'll jump around that block, no problem.
Well, the Cas9 could mutate.
Yeah, I guess that's
true. And that would be the end of it, right?
All true.
Because there's no reason, there's no selective pressure to keep Cas9 in these mosquitoes.
Right. That's true.
And so if it got a single mutation that it destroyed the endonuclease activity, that'd be end of the line.
Look at
this.
this.
Let's keep putting this in.
Nevertheless, Tony says here, this is worth ending on this.
Yeah.
Gene drive systems for population modification of vector mosquitoes have been proposed for nearly half a century.
That's right.
The phenomenal rate of allelic conversion achieved is a milestone achievement in the development of population modification strategies for controlling malaria and other vector-borne diseases. These efforts justify a degree of optimism for future successful application of this technology. We're fully aware that more work needs to be done, though.
Yeah, well, everybody understood that one.
But I share the optimism.
I mean, if you could do this on an island like Trinidad or Jamaica, where you're not worried about influx.
Yeah, but boats and travel. I mean, how do you keep mosquitoes?
No,
on
but
an
I think
island?
If you release these in
periodically-
India, Dickson, will some of them come to the U.S.?
Eventually.
Do we have this Stevens eye?
Stevens, I know we don't.
We don't.
It's not an invasive species. It's not. Unless you're looking at escapees from laboratories that work on malaria in the United States using Anopheles Stevens eye.
Has that been documented?
Yeah, plenty of times.
Okay.
You know, the insectaries, the doors open, everybody's complaining because they've got insect bites in
Daniel's
their
right.
ankles now.
The mosquitoes don't obey country borders, right?
They don't. No, but every time there's a new invasion, it pops up on PubMed.
No,
So it's an unusual
Yeah.
situation.
No, we always talk about Hawaii, right? Hawaii had no mosquitoes, right?
Come on,
and
man.
a ship, Captain Cook was able to travel all the way. How long did it
Totally
take him
agree.
to get there?
No, Hawaii is...
And now, we're hours. We're talking about a matter of hours to go from India to the United States. This is a 17-hour
Dickson,
direct flight from Delhi.
Is there a mosquito-borne disease in the U.S. that would warrant such intervention?
Well, we could talk about a lot of them.
I mean, West Nile is not that much, right?
That's not a great one to pick. I think
What's more?
Eastern equine encephalitis is a very dangerous.
Yeah, but not so many numbers, right?
No, but it's not about the numbers in that case. If it was your kid that caught it and died, I think you'd be right on point for this.
I'm not sure you'd want to modify all the mosquitoes for...
In the whole country.
No, you wouldn't.
I mean, for that, you need a vaccine.
But I want to say something else here, too.
Yeah.
It's not just a matter of introducing the vector into a new place.
Mm-hmm.
You have to match the ecology of what that vector requires in order for it to survive. And if you go to these microenvironments where they're from, it's very difficult to find a similar environment somewhere else. Very difficult, which is why they're found there to begin with. So that's part of the interaction between the environment
But
and
some, as you
evolution.
said, some species are invasive, like
Some are.
Albopictus, right?
Correct.
So that came to the U.S.
It
in
did.
tires, and
It
it
did.
spread.
It
right?
did. It did.
So,
And Aedes aegypti, too.
Yeah,
Aedes
what about?
aegypti
I was
is an
wondering about
invasive
modifying
species.
ticks for Lyme disease, right? What about that? We have 30,000 cases every year in Long Island
all
alone.
true.
That's a
yeah,
lot.
that would be good.
That wouldn't be bad.
So if there was a tick that could not get Lyme disease and we put it in there and it would drive it spread, the whole tick population in Long Island can now no longer get Lyme disease.
Ticks are really slow
That would be good stuff.
reproducers, though.
Is that worth the risk? And all we're saying is, eventually it might not work anymore. Well, still, during that time, you'd be...
They don't reproduce at the rate that mosquitoes do.
Maybe you would get giant ticks that would eat New York.
Yes,
Tick
what
Nate Westchester.
scary things will happen?
A lot of people
I can see the
feel
next
badly
horror
about
movie.
Yeah, that's right. It would be a horror
Wait a
movie.
minute. There is a superhero called the Tick, isn't there?
I'm not aware
Yes,
of it.
there is. If you go online right now, you can find it. It's one of the
Are you
Marvel
serious?
comic, The Tick. Yes,
Tick?
you bet.
Oh, Dickson. I'm surprised. Yeah, there is a guy named the Tick.
You have to go online to find out the veracity of my
Yeah,
statements.
in fact, I do.
Yes.
We have ample.
I'm hurt.
We have ample precedent. The Tick is a fictional character created by Ben Edlund in 1986 as a mascot for the New England comics chain of Boston area comic store. He is an absurdist spoof of comic book superheroes. Okay, the Tick. Yeah, with a name like that.
Yeah.
Call the Tick.
That's right.
He can help us. You have a case for us, Daniel?
Oh, I do. I have a great case.
They're all good, aren't
all
they?
great.
As Wink says, they're all good.
Thank you. All right, let's see. Okay, here we go. Now, this was a case. We have here at Columbia something called Morning Report. And what they do is if they have an interesting case come in, they invite one of the attendings to, sort of go through the case with them. And they feed you little bits. So I was sort of at the other end of this just a week or two ago as they presented this, what I found an interesting case. I felt like they gave too much information. So I'm going to sort of feed the information slowly. But here is the story. We have a 27-year-old female. She's a native New Yorker. And she was referred to an outpatient clinic here at Columbia after she had seen her OBGYN and told the OBGYN that she had seen worms in her stool and underwear. She said that these worms were about an inch or so in length. They were pale white in color. And she reports about 2 to 3 weeks of abdominal bloating. And I'm going to let you guys...
Dickson is shaking his head.
That's it? That's all you're going to tell us?
No, I'm going to let you ask some questions. We're going to go...
All right.
And that's how they do it, right? They give you a little
They do.
bit.
They do.
So she's pregnant, is that right?
She is not pregnant.
Okay.
That's a good question.
Just a routine OBGYN visit. Okay. She's 27. Sexually active?
She is sexually active.
What kind of diet does she have?
She just eats whatever everybody else eats around her. She's not particularly discriminate. She would say adventurous in her eating habits.
Tell me give me her description of these worms again.
She says the worms are about 1 to 2 inches in length.
Go on.
They're pale white in color.
Go on.
And she has...
What shape are they?
So they are long. They're longer.
Shape.
Longer than you want to know, like flat versus round.
Probably.
So interesting enough, she did say they were round.
Did she? Know?
Yes, she said they were round.
White.
Round worms, an inch long, white, pale, moving.
Moving. They do
An
move.
inch. An inch. That's a pretty big worm there, kiddo.
An inch.
An
I
inch.
actually, I have to admit,
An
I get to see one of these.
Wait, I must ask you another question. Does she understand what an inch is?
Yes, actually, an inch. I think an inch was accurate.
An inch was accurate.
Has she,
Having actually later on, I go on to actually see one of these, which is great, right? I get to see one of these moving, so I get to actually visualize.
So you verified the movement.
It is actually an alive, moving, unless it's an inanimate object that could move. But no, it's actually a 1 inch long thing.
You verified her description.
Yeah, I verified.
In all aspects?
No, not in all aspects. Later on,
we'll get to the differences.
Has she traveled lately?
Oh, yes, she has.
In my vast experience.
She is actually, she works for an NGO, and that's a non-government organization, right? One of these non-government...
highly underpaid.
She does stints working in a refugee camp down in the Ethiopian-Sudan border.
Really?
And I should note, looking at where these camps are, this is southern Sudan.
Very much.
Because people always say there's two Sudans. There's northern Sudan and southern Sudan, and it does matter. And so this is southern Sudan with the border of Ethiopia.
Right.
And she goes there and she works at the refugee camps.
And when was her last visit there?
She's been back there a couple times. And the last time, let's see if I have that, it was about a month ago.
A month.
And these worms appeared after she got back?
She noticed them when she was back.
Not while she was there?
Yeah.
She didn't notice them while she was there.
So when she got back from her last trip?
True.
Okay.
And as I mentioned, when she's there, she eats what the local people eat. She eats what people in southern Ethiopia, southern Sudan eat.
Any raw food?
There is a particular raw food that's quite popular in Ethiopia.
And what's that?
It's been in the New York Times, right? She likes to eat kitfo. Have you ever heard of kitfo? Kitfo is like a raw, like a steak tartare, and they serve it with melted butter.
Melted butter.
And it's quite popular.
And she likes to eat that.
It's very popular to take your dates to a kitfo shop.
Okay.
And share a meal there.
And this is beef. Do they grow beef in Sudan?
They do.
So it's local beef.
They grow also goats and sheep.
And I should mention also that she did not take malaria prophylaxis.
Mm-hmm. Did not.
She did not avoid swimming in fresh water.
Didn't involve.
But she did wear sandals instead of walking around barefoot.
Good for her.
So she swims in local water. Right?
She doesn't avoid it.
It's hotter than H.
She does wear sandals.
She does, yes. She wears sandals. She avoids walking barefoot.
But she avoids walking barefoot. Of course, she doesn't want to catch one of those parasitic fleas.
Yes, good. She listened apparently to the Tungiasis episode. No, yeah. I do not know that.
Where did she go to college?
Oh, I don't know. I don't know.
Anybody else travel with her?
But she is a college graduate.
Did anybody else who was with her have the same thing happen to them as happened to her?
She did not. No, she does not report that.
Why is she concerned about this?
She's a little concerned that there are these white worm things coming out of her
stool,
tail, and into her underpants. And
does she have any pain?
Well, she does mention 2 to 3 weeks of abdominal bloating.
That's right, bloating.
Abdominal bloating.
Is this continuous bloating or intermittent bloating?
It's like a constant, just distended, feeling bloated, uncomfortable. She does not have fever
or pain. So
and I should say we can, sort of the family history, nothing remarkable in the family. On exam, completely normal exam.
And these items
remarkable.
were, because you saw one, obviously,
Yeah.
available for diagnostic evaluation. They were sent to the lab.
Yes. One of these was sent off for evaluation.
Superb. So I was about to say that before, in my vast experience as a teacher of parasites and as a observer of parasites, I have never, ever encountered a white worm an inch long in stool that's round. Ever. So when you think about the common one, when you think of the common ones, we probably shouldn't go through the differential diagnosis now.
We could talk a little, and I'll just give you the last thing I think I'm going to give you is her lab. So she had a complete blood count. She had basic metabolic tests. She had liver tests.
Right.
I'll say they were all normal. She had a normal white count. It was not elevated, and her eosinophils were 2%.
2%. That's perfectly normal. So pinworm, which would be a candidate for diagnosis here, is a much, much smaller worm.
Not an inch.
No, it's much smaller than an inch. So you can't mistake it for an inch-long worm. If you confirm the size of the worm, Dr. Griffin, then obviously we can rule out the most obvious first diagnostic element, which would be pinworm. And she's an adult, so that would probably rule it out a little bit as well because they usually don't acquire it. But under stress, they could. So this is a conundrum. You've created another wonderful clinical conundrum. Maybe we should call this section Clinical Conundrums. Because
does she have any pets?
She does not. She does not have any pets, but there clearly are animals where she is at the refugee camp.
Right.
Yep. How is her stool? Is it normal?
No, she actually says her stool is not completely normal.
Meaning?
It's sort of loose stool, not 100% normal.
Mucus?
She's got sort of this bloating, not feeling so well. She doesn't describe mucus or blood in the stool.
But did you collect a stool sample?
A stool sample was actually collected and sent off for testing.
And what was the appearance of that particular stool sample? Do you recall?
I don't recall.
Don't recall.
I didn't actually physically see the stool.
Okay.
Did it hold the shape of the container?
Well, very interesting stuff. Raises a lot of questions, doesn't give a lot of answers because you have more data to supply once we get involved in this.
Once you move on further, we're going to look at that thing.
I'm sure we are.
We're going to look at that thing and see what it is.
So, Dickson, if you see a photo of this worm, you would be able to ID it immediately, right?
If it's a worm, you bet I would.
If it's a worm.
I have seen all the worms I can possibly see. You can't stump me with a worm.
You're a worm guy.
Well, you could stump me with a parasite from an animal that I've never seen before.
What do you call a person who studies worms?
A helminthologist.
Helminthologist.
That's correct.
That would include round or flat worms?
Or even segmented flatworms. That's correct.
Helminthologist. So are you a helminthologist?
I am. By training, I am a helminthologist.
Interesting.
But that's interesting because you don't call people who study Caenorhabditis elegans helminthologists, but they probably know more about worms than anybody. The genetics of Caenorhabditis has been well documented now, and so they have at the epicenter all of the basic information that you would need to put onto your system. And by the way, that's the way I became friendly with Martin Chalfie.
Would you consider Bill Campbell to be a helminthologist?
I think in his early days, yes.
Okay.
But in his latter days, he was a drug screener and interested in therapeutics.
Okay, are we done with that? Shall we move on?
I think that's enough for now. Yeah. People can write in with their differential, their thoughts.
By the way, this was a wonderful differential that was done for the last one. I mean, we had a spectrum of parasites that were estimated to be the cause of this woman's disease, and it turned out to be, of course, only one. But look at how many lookalikes there are, and the things that you have to go through to think about these things, right?
Yeah, and let's encourage people to put a differential in.
Oh yeah,
and not to just limit it to one. I mean, could there be a couple things going on in this woman that maybe is
good idea.
So try to explain. We've got bloating. We've got this thing in the stool. Is that two different things causing it, or
confirmed by a physician?
And this is confirmed. So
all right.
So we actually do know the diagnosis, and let's see if people can give us some thoughts on what it might be.
Exactly right.
All right, let's read a couple of email.
Okay.
Okay?
Yes.
First one is from Hans, who writes, "Hi, TWiP team. Writing from Seattle on a warm July night." Wow, this is an old email. "Walking home from a long evening of transfections and binding assays. Just finished listening to TWiP 73 about E. histolytica trogocytosis. Very excited to hear you covering that paper, as I actually started my science career as high school and then college student in Bill Petrie's lab at the University of Virginia." You know Bill Petrie?
I do.
"I'm now at the University of Washington working on maternal to infant HIV transmission."
It's a good place to work on that.
Would that be, what's her name?
Yeah, that's right. I would think of that woman too.
Also applying to MSTP programs, and enjoyed Vincent's TWiV interview with the group of MD PhD students at Vanderbilt. I'm writing because Dr. Despommier seemed quite convinced that E. histolytica trophozoites needed to be cultured in the presence of feeder cells. While I can confirm that trophs are quite excited, forgive the anthropomorphizing, to gobble up most anything you feed them, it's not necessary that they be around in culture. We use a chemically defined media similar to LB. It's a broth that you use for growing bacteria. Luria broth, right? Luria bertani, actually. But with different components. And, of course, we grow under anaerobic conditions. These cultures can be xenic or axenic depending on the isolate and application. Just wanted to clarify that point and tell you that I love listening to all of your podcasts in lab. Keep up the good work, and thanks for giving E his some attention. Yeah, some attention to E, right?
Well, I'm grateful for the update and the improvement of the data. It's nice to see that they've conquered the in vitro cultivation.
Dickson, you can read Blazes.
I shall. Blaze writes, "Yes, yes, yes to Colbert's show. And I have the perfect pitch for the showrunner. Remember how Jim Fowler would always bring animals on to the Tonight Show? Always a popular segment. Dickson and Dan bring live parasites to the Colbert Show. Imagine the screams, live ascaris, writhing tapeworms. Put together a pitch and email that sucker to Colbert today."
Could you get live worms to bring on the Colbert Show?
I don't think that would be a problem.
Really? You could buy them from some biological
No, you
supply
can go to a slaughterhouse and get live ascaris, but they're ascaris sum. They're not ascaris
That's okay,
parties.
as long as they're writhing.
I want to go back. Can I please go back to one of the guesses on our last thing? Because they thought it came from a dog. Ascaris ricoides can't infect dogs. It does not infect dogs. So that was a little bit of a disconnect there. I meant to add that to our diagnostic.
Who said that? Let's see.
There was a woman that said that. And
is that Elise, maybe?
Maybe.
Well, that was her guess.
But the dogs can't be looked at as a
local dogs. Yes, she did say
that can't be
impossible.
Right. No, impossible.
Okay,
but there is a Toxocara canis, which is their ascaris, which can infect us as a worm larva. But that wouldn't give you the liver symptoms that we were talking about. So,
all right,
let's just correct.
Daniel, the next one up is Layla.
Layla writes, "Hi, Vincent, Dickson, and Daniel. I am a postdoc, and I've listened to TWiP off and on for the last 5 years. It has always been my favorite of the Twix podcasts, but I've enjoyed it even more with the addition of the case studies. I finally thought I would try a diagnosis of the case study. Google was absolutely necessary as my degree is in molecular biology, not medicine. I narrowed my guess to tapeworms or roundworms based on symptoms. I think I'm going to go with roundworms ascariasis as my final diagnosis, since there are descriptions of humans passing these worms through multiple body orifices, as opposed to tapeworms, which seem to be primarily restricted to stool." Do you know which episode she was answering on?
Not. Let me see if I can do a search. Lela. Here we go. TWiP 91.
Wow.
Okay, what was that?
All right,
one?
let's look that up. Microbe.tv/TWiP.
While Dickson looks that up, I'll continue reading.
Nice.
I also
Actually,
want
Vincent's
to
looking
mention
that
a
up.
science fiction book I read a while back, Symbiont by
Oh.
Maria Grant. It is actually the second in a series about tapeworms that were engineered to secrete medicine to treat diabetes, etc., but managed to migrate into the host brain and either become zombies or, in a few cases, integrate into a tapeworm/person. I thought just the idea was pretty awesome, and it is good summer reading. Thanks for the show. I enjoy listening and telling everyone about the cases, Leela.
You could end up with a segmented personality.
Dickson. Okay, TWiP 91 case study was, let me scroll through all this email here. TWiP 91. That was a long time ago. All right. This was the 28-year-old Utah internal medicine resident presented ID with worms. He vomited them out.
Yeah, that was Ascaris.
Okay.
Yeah.
Yeah, it was Ascaris.
And he traveled to India, etc. Okay.
Yeah.
Did you finish that one while I was looking? Symbiont.
We finished.
Is it my turn?
Caleb writes.
Caleb writes, greetings, Dr. Racaniello, Despommier, and Griffin. It is a chilly 66 degrees F today in sunny Riverside, California. I hope all is going well, and you will be twipping again soon. I discovered your podcast approximately 2 months ago in the search for something much more entertaining than listening to NPR, yes, or other such talk radio on my way to and from my university. I am currently a first-year PhD student in medical and veterinary entomology at the University of California, Riverside, and these podcasts have been absolutely amazing. I listen whenever I get the chance, walking around campus to get coffee, taking care of my house flies. I'm working on looking at the genetic mechanism behind behavioral resistance to insecticides in house flies, and every chance in between. I listen to the most current 10 podcasts or so very quickly, seeing as I can listen to them at 1.5 or 2 times speed and cover the information very quickly. I picked up this habit from my girlfriend who is in medical school. Well, I hope she's learning.
Indeed.
I'm quickly making my way through the backlog of episodes. I'm currently on 53, anti-saliva immunity.
Cool.
These podcasts have made my passion for parasitology and vector-borne diseases grow exponentially.
Neat.
I got involved in medical and veterinary entomology due to a wonderful professor I had in my undergrad named Bradley Mullins, who teaches medical and vet entomo course at UCR. I wonder if any of you know him.
Sorry, I don't.
I know that he gave a plug for Dr. Despommier's book, People, Parasites, and Plowshares, in one of his lectures.
Oh, great.
But getting down to the real reason I emailed is that last week I saw a very interesting paper talking about genetically modified mosquitoes who are resistant to malaria.
My
In
goodness.
quotes. I skimmed the paper and found it fascinating, just having learned the ins and outs of CRISPR, Cas9, RNAi, etc. Then listening to TWiP last night, I listened to the episode from 2 years ago where you had Dr. James on talking about some of his work. The timing was impeccable. I just wanted to let you all know to keep up the great work, and I am actually considering having my minor for my PhD be in parasitology.
Oh, great
thanks
idea.
to the amazing content you have put out over the last few years.
Great idea.
Now, he is going to love this episode.
It's perfect.
It's his paper.
You
All right,
bet.
let's do one more round, Dickson.
Okay. Paulette writes, 99 case study. Hello, Doctors Racaniello, Despommier, and Griffin. Dr. Griffin's case this episode, characterized by an itchy black-centered nodule lesion on the toe of a beach-combing tourist recently returned from Brazil, suggests that the toe bug, Tunga penetrans, hitched a ride with our innocent victim back to the States. A tiny sand flea is a small price to pay for enjoying the beautiful beaches of a tropical paradise. But I will stay home myself. Yours, Paulette and Bob.
Oh, should I keep reading? Hi, docs. Here's a YouTube, and we see a YouTube link here. Someone sprays a giant mantis, and as it dies, out comes a worm that is twice as long as it is. He asks whether or not it's a round worm.
Love to see a picture.
It's a movie, Dickson.
It's a movie, Dickson.
Well, I don't know why they're spraying a mantis to begin with.
Exactly,
Mantises
right.
are good, and I feel very badly that this person first
tell me
knocked
about
it with
this.
a stick and then sprayed it.
Tell me about
However, Dickson,
Maybe they didn't know it was a mantis.
how can you not know what...
Well, some people don't know about these
Anyway,
things.
there's a long, black, thin worm coming out.
Yeah, it might be a myrmithid.
Oh, my God. So it wants to get out when the thing is dying. I really feel badly that he killed or she killed this mantis.
Right. That's
Yeah, look, he kind of got it with the stick
sad.
there.
That is sad.
So if you go down to about 1 minute and 15 seconds, you'll see the worm coming out.
Okay, we'll move
It's longer than the length of the mantis for sure.
Yeah, I can actually see that thing.
Oh, there it comes.
Oh,
there
my
it comes.
gosh.
Poor mantis.
Yeah, I would say that's a myrmithid. There's two of them.
Myrmithid?
There are two of them.
No, I think that's shadow that you're seeing below.
Oh, okay, fine. Yeah, you might be right.
You're a shadow of your former self.
Always have been.
Oh, my gosh, look at that thing.
Yeah, it's
quite. What is it going to do now that it's out, Dickson?
It's going to die.
Because it needs another host, right?
There you go.
Can it get into another host at this stage?
Probably
its life
not.
cycle?
I think it gets in as a larva of a quarry that the predator, in this case I am absolutely right about the predator nature of mantids, he ate something that he wished he hadn't.
This has 3.3 million downloads.
Wow.
Oh my gosh.
Apparently that is referred to as a Satan worm. No, I'm just saying from the comments below.
Well,
that is what you should hit with the stick, not
my
the mantis.
guess is that if you looked up the word myrmithid parasites, you might find them.
Satan worm, that's right.
Yeah, but why? I wonder why the person knew to whack this thing and then
I don't
have
think
his
they
camera.
knew, but they just took a chance.
They didn't know.
And we're able to keep the camera nice
I'm
and
very
steady.
sad that he killed the mantis.
Yeah, that's one of those things you do not do, people.
No.
That was yours, Daniel, right? Let's take the next one.
I'll take the next one. Peter writes, "Greetings, Dickson, Vincent, and Daniel. Here are a couple of recent papers that I think would be worth some discussion." And researchers at Imperial College London have engineered Anopheles gambiae mosquitoes using CRISPR-Cas9 to pass on genes that cause infertility in female offspring.
Oh.
Actually, this was an article that my 15-year-old daughter, had read, and we were discussing. But it's a CRISPR-Cas9 drive system targeting female reproduction.
Yeah, the same idea of what we did today, basically.
But you estimated that that's not a good idea because then it would die out before it got a chance to perpetuate.
Well, it's a drive system.
It's a drive system.
It's like the one we thought. It's a Cas9 drive system, yeah.
And the other paper...
I'm sorry, Dickson, would pass... I'm sorry, Daniel. To pass on genes that cause infertility in female offspring. Female, but the males would have it, and they could keep mating until they wiped out the whole mosquito population. Would that be bad, Dickson?
That would be great.
If we took all the mosquitoes from the earth?
No. This is just Anopheles gambiae.
That would be fine.
Anopheles gambiae, which is the premier vector for transmitting malaria in Africa.
Daniel, would you be okay with wiping out a mosquito species?
No, I hate to say. I mean, on some level, I would, but really? I don't. I think from the environmental standpoint, I would be concerned.
Just one species?
They're pretty important. I mean, we already have issues in parts of the world with pollination. I mean, mosquitoes don't just bite and spread malaria.
Let me tell you,
pollinate. They're a food source.
The human species has eliminated more species of things in the world than just one species of Anopheles gambiae. We eliminate all kinds of useful things.
I'll go out on a limb and say I still don't think that's okay. I don't think that makes it right.
At the public discussion time, you're allowed to say whatever you'd like.
Yes, let's stop extinguishing species and start getting some more on the list. Right. The other paper is based on 9 years of longitudinal data from 986 Semaine forager horticulturist women living in the Amazonian lowlands of Bolivia.
Right.
This indicates that women infected with Ascaris lumbricoides tended to have an earlier first pregnancy and have more children than uninfected women or women infected with hookworm. Hookworm infection caused a reduction in fertility. The increased fertility is thought to result from systemic immunological changes caused by Ascaris lumbricoides. Now, I will say, so this journal, helminth infection, fecundity and age of first pregnancy in women. I thought there were a lot of confounders, a lot of speculation.
Exactly.
And I was not convinced that there was a causal relationship here. It almost seemed like women who have earlier first pregnancies and have more children, may be in an economic situation where they're more exposed and more likely to be infected.
Right.
It was sort of my thought, maybe more of a marker than a true causative relationship.
All right, we have two more. Let's do them both. The next one's from Jesse. Hello, Twippers. I can't believe I'm writing for the first time. I've listened to TWiP, TWiV, and TWIM off and on for the past few years. I will say that as an internist, first in Seattle and now at LA County Hospital, TWiP is ironically my favorite, as it has historically been the most clinical of the three. I say that this is ironic because, of course, my day-to-day practice is all virus not otherwise specified. HIV, HSV, Staph aureus, strep pneumo, E. coli, Klebsiella, Pseudomonas, etc. I will only occasionally encounter a parasite, usually toxo in an immunocompromised host, although I know well that it is possible that many a parasite pass by me unrecognized. I will further add that I think some of the early episodes of TWiP, where Doctors D and R elaborated on the life cycles and clinical aspects of some of the classics, malaria, for example, are some of the greatest podcasts I have ever heard. Thank you. So good were those episodes. I even remember where I was when I listened to them, running up and down beaches in the Dominican Republic with shoes on, I hope,
right,
where I was attending a wedding. And this is high praise because I listen to a lot of podcasts. Of course, sadly, this is why I can't listen to every episode of TWiP, which is quite long. If I listened to every episode and the Slate Political Gabfest and the World Next Week and so on, I'd have to quit my job and leave my girlfriend. I would do the latter. It will only cause you problems the rest of your life.
Exactly.
There are also all the medical podcasts I listen to. I wonder if you have heard of Dr. Mark Crislip's infectious disease podcast. There's one called Puscast and another called A Gobbet of Pus. Both are fun and interesting.
Love it.
I have listened to them. Okay, I've been known to go on and on. It's already too late to stop that, but I'll try to wrap it up. First, I am so glad you all talked about the H. Nana case. I anxiously awaited this last episode with the expectation that you would, and the discussion did not disappoint.
Great.
Second, I heard that you offered to send a PDF of Dr. D's parasitic diseases, and he goes on talking about that. I sent it to him, so that's done. And I almost forgot, the weather here in L.A. is no weather at all. I think W.H. Ouden said that. Being from Chicago, I miss the bitter cold. People think I'm crazy, but I do. Thank you for your podcast. And Dr. R, between the podcast and your job at Columbia, I have no idea how you do it. You must have infinite energy. I am jealous. Sincerely, Jesse. P.S. I'm looking forward to This Week in Fungus. I would call it The Fungus Among Us. How about that?
Good.
I love fungus for some reason. We see it in our most immunocompromised patients, and for whatever reasons, I'm always fascinated. I've diagnosed mucor, which is the term we use to refer to a whole host of invasive molds, including mucormycosis itself a few times. It's always just terrible, with the patients needing surgery, about 10 different antifungals, and prayer or luck, depending on your belief system.
Right.
But I think I find it fascinating because it speaks to the fact that without an immune system, we would all just be overrun by fungus. Maybe the most successful species or family, or who knows of them all. I highly recommend you look up Oncology versus ID on YouTube. It is a good and hysterical commentary on this. Wow. Do you feel that's from a co-physician, Daniel, the way he's writing?
Yes, I felt a camaraderie there, and and unfortunately, we had a unfortunate case of a woman with mucor in the clinic today, who, thanks to her insurance, was having trouble getting her antifungal medication, and for 4 weeks now has not been treated for her mucor.
Oh dear!
Really horrible. Yeah. I don't like funguses. I mean, I find them fascinating, but I also find them rather frightening.
Fungus is a kingdom, by the way.
Indeed it is.
All right.
Indeed it is.
When is along with plants, animals, protists, and bacteria. Right, Dickson? Am I right?
You are correct.
Dickson, can you take the last one?
Well, I'll try. Bryce writes, "Hello, TWiP team. I was first introduced to this podcast by a fellow lab member. Needless to say, I was hooked by my first episode. Then imagine my excitement when I found out that Dr. Despommier was a Notre Dame graduate, an alumnus. Go Irish! Currently, I am in a Notre Dame lab investigating the ectoparasite Culex quinquefasciatus. Ectoparasites and mosquito species, by the way. The passion of the TWiP team and guests is infectious. However, much to Dr. Racaniello and Dr. Depommier's disappointment, I will be pursuing an MD in the coming years. That's not a disappointment. Therefore, Dr. Griffin, do you have any words of guidance for a future MD who wants to remain in tight association with parasites and research? Thank you for your phenomenal podcast. Best, Bryce."
I'll throw you some advice, Bryce.
This is Bryce advice?
I don't know if it's that essential during the medical school years, but when you do go on to do further training, if you're going to be in a coastal, major urban setting, you're going to see more parasitic disease, more tropical medicine. And particularly if you sort of push that way and get a chance to do some overseas work, you'll get to see your share of parasites. But, you're going to have to be proactive. You're not going to necessarily see it. You're not going to see a lot of it if you end up in Ohio, Arkansas, other sort of central.
Fort Collins.
So Fort Collins is a little unique. I think there's just, if you go to a place like Utah, or Fort Collins, but there's a lot of travelers. People are coming back. So either places where there's a lot of incoming immigrants, that's really the best. Travelers, second best. And getting overseas is probably the best.
And I would add, join ASTMH, the American Society for Tropical Medicine and Hygiene, and you can keep up with the literature and go to the annual meetings, and you can meet a lot of docs that are involved in foreign travel and diseases, exotica, et cetera, et cetera. And go Irish is right.
But, of course, the most important is to keep listening to TWiP.
Listening. Just keep
if at night, if you
TWiP.
can't fall asleep, put those earbuds in, turn it on, and you'll be sleeping in no time.
Well, I got my inspiration from people that you obviously are talking to, too, because I can only relate the enthusiasm that I acquired from others that had it before me, and some of them were at Notre Dame. And so it's a great school, and it's a great place to be from, as well as to go to.
And hopefully the clinical vignettes Bryce will give you a little leg up when you start doing your clinical. You'll have a sense of the presentation, and maybe a few of these you'll end up seeing.
You can outshine the house staff.
Make us proud, Bryce.
There you go.
Well, those are wonderful emails, right? A lot of people are inspired, and that's great. That's what we want to do as teachers. Yep. And this is TWiP. You can find TWiP at iTunes. You can also find it at our new home, microbe.tv/TWiP, where all our other podcasts are housed. I was going to say hosed, but they're not hosed. Housed, including TWiV, TWIM, TWiVo, and urban agriculture, and more to come. And in fact, if you do a science podcast and you'd love to be part of the Microbe TV team, drop us a line. What was the email? That shows@microbe.tv, shows@microbetv.tv. And, of course, we love getting your questions and comments. We have a new email address as well, twip@microbe.tv. New year and a new email, twip@microbe.tv. And it should stay that way until I am deceased. Not going to change it again. Daniel Griffin is right here at Columbia University Medical Center, where he does the important stuff. Thank you, Daniel.
Oh, thank you.
Looking forward to more. And Dickson Despommier is at Racaniello.org. Thank you, Dickson.
You're welcome, Vincent.
Thank you for 100 episodes.
Yeah,
you and I, we did every one.
This does not seem like 100, does it?
No, it doesn't. It's not work.
It's not work. It's pleasure. And education.
I hope we can do many more.
But, as long as I'm breathing in and out and can speak, I'm yours.
What if you start forgetting things? No, you already have.
You'll notice that. No, that's not, well, I haven't reached that point yet, but I'm presuming that eventually with age, everybody reaches that point.
Our listeners will jump in.
I have a ton of problems with names. People's names. I know that a lot of people will agree with me out there that are past 50 years old. You start that Rolodex turning along in your head. You try to come up with the name of somebody that you've known forever, and it just doesn't come up right away. You have to start with the alphabet and that sort of thing. So that's just part of the aging process that I have to accept. And I hope the listeners do, too.
You seem to be in good shape, so you're probably good
fine.
for a few more...
I feel like I'm still in control of my faculties.
You're probably good for a few more weeks.
I'm good for a few more weeks?
Yeah.
That's not a quite...
Oh, I think you got at least a month left.
Be nice to Dickson, remember?
Actually, it was quite nice this episode, wasn't I? That's all you're going to get this year.
That's it. I've used up my quota.
If we do this every 2 weeks... that's 25 episodes this year.
Yeah.
And if we do roughly 10,000 downloads, we'll hit 1 million this year.
Oh,
easily.
I'm sure we will.
Easily. That will be fun.
That'd be great.
All right. So I would like to thank you both for doing this. I really appreciate it. I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.